The first layer of the cutaneous membrane that we're going to discuss is the epidermis. And so epi meaning above, above the dermis. So the epidermis is the most superficial layer, and we'll see several different cell types within the epidermis, but the majority of the cells, 95% of the epidermal cells, are made up of this specific cell type called a keratinocyte. And keratinocyte's main function is going to produce keratin, which is a tough fibrous protein. So remember, epidermis, we talked about this in chapter 4. We find stratified squamous epithelium that makes up the epidermis of the cutaneous membrane. Lots and lots of layers of squished, stratified squamous type cells. This is so that we can have a mechanical barrier. We can have lots of cells. The cells towards the apical surface can slough off, and we still have cells left behind. So it makes for a good barrier. Anything that's open to the outside and subject to abrasion, that's stratified squamous epithelium. Okay. So these keratinocytes, which make up the bulk of the epidermis, main function again is to produce keratin, which is a tough fibrous protein. These cells will also be linked together by an inter meaning between two cells, so intercellular junctions called desmosomes. Remember, we talked about desmosomes in chapter 4. These are these huge complexes that have an extracellular component that are going to connect the adjacent cells together. And then this big intracellular component, which the proteins of the desmosomes actually attach to intermediate filaments of the cytoskeleton, so they really anchor the cells together so that they can withstand shearing forces. All right, and so that's those two things alone, the fact that keratinocytes produce lots and lots of this tough fibrous protein called keratin, and then the fact that they are linked together through these desmosomes, and then also the fact that there are lots of layers, multiple layers of these keratinocytes, all help give the epidermis its function of being a tough mechanical protective barrier between the external environment and the internal environment of the human body. So we're going to talk, so within the epidermis itself, it's 95% are keratinocytes and it's a stratified squamous epithelium, but there are specific layers or strata within the epidermis. And so this is just all typed out for you, but I'm going to actually use this picture and talk about each of these strata, and what the cells are doing, how they're functioning in each of these layers. So to orient ourselves, what we're looking at is up here would be the apical surface or the outside. And then down here, this would be the basement membrane, which is going to anchor this stratified squamous epithelium of the epidermis to the connective tissue below. So all this stuff down here, this would be the connective tissue of the dermis. And then here, all of this is stratified squamous epithelium of the epidermis. And so there are layers or strata within the epidermis, and each have their specific function. All right, so the first layer that we're going to talk about it from the deepest layer, which is right adjacent to the connective tissue. We're going to start with this layer and work our way up to the most superficial layer of the epidermis. So this first layer, it's a single layer of cells called the stratum basale. Stratum basale. So basal, or you think basement, it's this single layer of cells that are anchored directly to the basement membrane. They're right next to the connective tissue of the dermis. So they're stratum basale. So these cells, if you look over here in the micrograph, they have somewhat of a cuboidal-ish shape to them, and that's because they are actively undergoing mitosis. So these cells the cells of the stratum basale are stem cells. They have the ability to reproduce, make over and over and over again, undergo mitosis to continue to generate more keratinocytes. And that makes sense, right? Because we talked about stratified squamous epithelium being, cells have to be able to slough off because they are subjected to lots of shearing and abrasive friction forces. So we have to have the cells slough off, but we have to, regenerate more so that we don't eventually lose our epidermis. So these stratum basale cells are actively undergoing mitosis over and over and over again, just making copies of keratinocytes. They're very metabolically active. Mitosis is very energy expensive, so it makes sense that this very metabolically active layer would be located directly next to the dermis, because remember, epithelium will not be vascularized. Epithelium is avascular, whereas the connective tissue, like the loose connective tissue that we find up here in the dermal papillae, and then the connective tissue down here, which is the dense irregular connective tissue, those layers will be vascularized. So these extremely metabolically active stratum basale cells are closest to the nutrient source, because these cells rely on diffusion of nutrients from the blood vessels in the connective tissue. So that makes sense. They're super metabolically active. They need a lot of nutrients because they need lots of energy so that they can undergo mitosis constantly. So they are closest to the vascularized connective tissue. All right. And notice that the epidermis is not just, the stratum basale is not just a flat layer of cells across, but rather we see these invaginations, these big folds of the epidermis that helps increase the surface area. One, we have way more stratum basale cells this way that have the ability to undergo mitosis, and then more surface area for diffusion of nutrients from the vascularized dermis up to these stratum basale cells. So the next layer is going to be the stratum spinosum, and this is going to be the thickest layer of the epidermis. And how I always identify the stratum spinosum is I look for the stratum basale first, which is the bottommost layer. So over here, stratum basale would be the layer of cells right next to the connective tissue. And then if I skip the spinosum and if I go up here, this layer of cells that are really squamous in shape, and they're very dark staining, right? They're dark staining because they have lots of cytoplasmic granules. This is the stratum granulosum. And so I identify the stratum basale, then I identify the stratum granulosum, and essentially all of the layers of keratinocytes between the basale and the granulosum, this is all spinosum. Okay, so all of these cells are the cells of the stratum spinosum, and they're called spinosum because they have under a microscope, when they were first being identified, they had a spiny shape to them, and so they were called the stratum. They were dubbed stratum spinosum. These are keratinocytes just being keratinocytes. So they are just carrying out normal keratinocyte metabolic activities. They start to take on much more of a squamous shape to them. Their overall function is really just to add to the layers of the epidermis. The more layers of stratum spinosum we have, the thicker the epidermis can be. So they add to the layers of the epidermis, and as these cells from the stratum basale migrate up through the stratum spinosum, they'll start to produce more and more keratin. All right, so they produce more and more keratin as they move up through the layers of the stratum spinosum. All right, but these cells, these keratinocytes, especially the ones that are down here and the more the deeper layers, these are still relatively metabolically active and mitotically active cells, and they can be because they're still relatively close to the nutrient supply of the dermis, so they can pretty readily get nutrients from the blood vessels of the dermis, so they can still be relatively metabolically active. Now, as these spinosum cells, so the keratinocytes of the stratum spinosum, as they move up towards the stratum granulosum, what happens now is that the keratinocytes of the stratum granulosum, they start to accumulate all of these cytoplasmic granules. And those granules are going to be keratin granules. So lots and lots and lots of keratin protein in these little granules, that tough fibrous protein. And the cells of the keratinocytes of the stratum granulosum also produce lots of a lipid substance, which gets secreted by exocytosis. All right, so the main function of the stratum granulosum is to produce lots and lots of these keratin granules and to secrete a lipid substance via exocytosis. Now that lipid substance will be hydrophobic, and it will coat these keratinocytes. So this lipid hydrophobic substance coats these keratinocytes of the stratum granulosum, and it's pretty much waterproofing the epidermis, right? So hydrophobic lipid substance means that water, anything that's polar or water soluble, can't get in or can't leave. So it's critical to maintaining water and electrolyte balance so that we don't absorb too much moisture or polar substances from the environment, but also we don't lose moisture from our internal environment. All right, so that's the function of the stratum granulosum. It just fills up with lots of tough fibrous keratin granules and then secretes that lipid hydrophobic substance that coats the cells and waterproofs the epidermis. But when these cells become coated with this hydrophobic lipid substance, it also causes them to not be able to get nutrients anymore through diffusion. So these lipid-coated keratinocytes can't get nutrients through diffusion, and if you look at them now, their distance from the dermis, they're even further away from the blood supply, so diffusion itself is limited, and then whatever nutrients could get all the way up here can't penetrate that hydrophobic barrier that surrounds the keratinocytes now. So what happens is these cells are far away from the nutrient supply. They're also coated with that lipid hydrophobic substance. They can't get nutrients at all, really. Very limited nutrients, and so they undergo apoptosis, and that's what this next layer is. So the stratum lucidum means clear, all right? The stratum lucidum is this layer where these keratinocytes are so full with keratin that they can't really function anymore because they're just so full with keratin, and then they're also coated with that lipid substance and they can't get nutrients. They're also really far away from the nutrient supply of the dermis, and so they undergo automated cell death called apoptosis, and they die. And so that's why up here you no longer see nuclei in the cells because they are dead, right? They've undergone apoptosis. That's what happens in the stratum lucidum. And then all of the cellular debris, as well as the tough fibrous keratin, accumulates and forms this big, thick, dead outer layer called the stratum corneum. Right? Like a corneum, a horn. It's the tough, fibrous, outer, superficial most layer that's made up of dead cells. It's pretty much just compacted keratin and cellular debris. All right? Now, because these keratinocytes are dead up here, remember we don't see nuclei, because they're dead, their desmosomes, those intercellular junctions, are also compromised. They're no longer really working because the cells are dead, and that enables these outermost cells, because these desmosomes have lost their integrity, these cells can slough off. And so as your skin comes into contact with, just rubbing up against anything really, when you put clothes on, when you dry off with a towel, when you scratch your skin, any sort of abrasive or rubbing forces, these dead keratinocytes that no longer have desmosomes can slough off, but we don't have to stress because we have lots and lots of layers behind. And even as you slough off more, guess what? That stratum basale is constantly undergoing mitosis, producing more keratinocytes. So every time the stratum basale produces more keratinocytes, they start their new life cycle. So this stem cell right here, this stratum basale stem cell, undergoes mitosis, produces a new keratinocyte. That new keratinocyte will work its way up through the stratum spinosum, just being a keratinocyte, producing keratin, being metabolically active, participating in the lots of layers of the epidermis. Then it reaches the stratum granulosum. It completely fills with keratin granules. It secretes a hydrophobic lipid substance through exocytosis, coats themselves in that lipid substance. Now we have these tough fibrous cells that are coated and waterproofed. They are very far away from the blood supply of the dermis, and so they die. They undergo apoptosis in the stratum lucidum, and then the leftover cellular debris and the keratin protein compacts and forms this stratum corneum, in which these dead cells can slough off, and that's the life cycle of a keratinocyte. All right. So this is just talking about some topical medications, what can and can't get through the epidermis. It mentions that some antibiotics or some medications might be toxic if taken by the mouth, but if you need it to have a local effect on the skin, right, you could just apply it to the skin, especially if it's a polar type substance. Polar molecules cannot pass through the epidermis very readily because of that lipid substance that coats those keratinocytes. And so these topical ointments don't have a really good chance of reaching the blood vessels in the dermis. So you have just a very local effect on the superficial epidermis if it's a polar substance that you're applying topically. Non-polar substances can cross the epidermis more easily because they can get through that lipid waterproofing substance that coats the keratinocytes. That's pretty much how birth control patches work. So birth control patches contain hormones, synthetic hormones like synthetic versions of estrogen, synthetic version of progesterone, and those are steroid hormones. So they're based off of cholesterol, which is a lipid. So that's lipid soluble. And then the coating around the keratinocytes is lipid soluble. And so these hormones can very readily or very easily pass through the epidermis and get to the blood supply of the dermis. And so that's how those hormones from birth control patches are administered. And then it just mentions down here, so things like heavy metals, a lot of times are non-polar. So heavy metals are very easily absorbed through the skin and can get into your blood. If you work in a lab and you work a lot with, say, like methanol or chloroform, those are non-polar substances, and so those non-polar chemicals can very easily get across your epidermis and can be fairly toxic to humans. All right, this is just a way to learn the layers, so it gives you this brilliant studying, gives loads of confidence. Brilliant, so B for stratum basale, down here, undergoing mitosis, very close to the blood supply, super metabolically active. Studying, the S means stratum spinosum, the thickest layer, lots and layers of keratinocytes, just being keratinocytes, starting to fill up with keratin. G, stratum granulosum, they're super duper full with keratin granules, so full that they can't really, operate very well. They're also very far away from the nutrient blood supply of the dermis. They also secrete a lipid hydrophobic substance that coats and waterproofs the cells, but then they die because of it. That's the stratum lucidum, L, and then the leftover cellular debris and keratin compact together to form the stratum corneum, C for corneum. So this is just a mnemonic to help you learn the layers. You should know the life cycle of a keratinocyte. Where does it begin? Down here in the stratum basale. What happens? What's the function of each of these layers as it reaches the apical surface and sloughs off? How do they regenerate more? So everything I just talked about, you should be able to explain in a short answer question the life cycle of a keratinocyte. And so that's this slide right here. Overall, the timeline for a keratinocyte life cycle is about 40 to 50 days from being formed through mitosis at the bottom layer, the stratum basale, to being sloughed off from the stratum corneum. This is just these two slides if you want to better understand how these cells migrate up through the layers. This just gives you a way to learn that, so you can read through these slides if you had trouble understanding it from me. So it's just a little concept boost from your book. All right, the other cell types that you can find in the epidermis, again, 95% will be those keratinocytes, which in this cartoon picture would be the pink cells. Right, so stratum basale, they're kind of cuboidal-ish because they're undergoing mitosis. The spinosum, stratum granulosum, we don't really see the lucidum here, and then the stratum corneum. All right, so dendritic or Langerhans cells. Langerhans is just the name of the gentleman who first identified these cells. They are represented here in blue, and they're called dendritic cells because they have these dendritic processes that come off of the cell body. These are phagocytes. So phagocytes are cells of the immune system that have the ability to phagocytize unwanted cellular debris as well as pathogens, so foreign pathogens. So not only is the epidermis is made up of lots and lots of layers of cells, which makes it more difficult for foreign unwanted pathogens to get through, right? But we also have another added layer of protection in that we have these phagocytes throughout the epidermis that if an unwanted pathogen were to be able to get through, and say this pathogen gets through these first couple of layers, well, then it comes across this dendritic cell and gets phagocytized before it can make its way into the blood supply of the dermis and maybe cause a systemic infection. So that's another line of defense against foreign pathogens that might be able to get through some of the layers of the epidermis. Merkel cells, these are specialized epithelial cells found in the stratum basale. They're represented here in red, but they are not stratum basale cells, so they do not produce keratinocytes through mitosis. All right, they're located in the basale, but they are not keratinocytes. There are specialized sensory receptors, and I like this picture because it shows you here, this is the Merkel cell, specialized epithelial cell that functions as a sensory receptor, and it's associated with a sensory neuron. So this is a sensory neuron of the nervous system, and it's going to, so what this cell type is able to sense is fine touch. So very fine touch, as well as it has the ability to discriminate textures and shapes. All right, so detects light touch and discriminates shapes and textures. So you'll find larger numbers of Merkel cells in areas that that are specialized for fine touch, like your fingertips, lips, and at base of hairs. So when you feel, feel your hairs kind of move and you can sense that, it's because of your Merkel cells. It makes sense that these Merkel cells would be able to detect fine touch because they're located in the superficial epidermis, right? Right, so they can discriminate very fine touch. All right, so they are a touch receptor. And then so we talked about keratinocytes, dendritic cells, Merkel cells, and now the fourth cell type that you find in the epidermis are melanocytes. They're represented here in brown. Melanocytes are also found originating in the stratum basale, but again, they are not keratinocytes. But the cell body of a melanocyte is located in the stratum basale, but they have these huge cytoplasmic processes that reach up and extend throughout the epidermis. And what a melanocyte does, so here, melanocyte cell body, and then these huge cytoplasmic processes that reach up throughout the layers of the stratum spinosum. Melanocytes produce a pigment molecule called melanin. And so it's a pigment, a protein pigment. It's what not only gives color, we'll talk about skin color a little bit later on in the chapter, But it not only gives us our skin color, but it offers protection against UV radiation. So a pigment is a molecule that has the ability to absorb certain wavelengths of light. And so melanin has the ability to absorb some certain wavelengths of UV radiation, and it protects us from damaging our DNA. All right, so this is another line of protection from the environment, from UV radiation in our environment. And so this is just another picture, a nice little cartoon picture showing all of the strata: basale, spinosum, granulosum, lucidum, and corneum. And then it shows you Langerhans cells, Merkel cells, and melanocytes, and where they would be located in the strata. Langerhans or dendritic cells are located scattered throughout the spinosum, Merkel cells in the stratum basale, and then the melanocytes also originate in the stratum basale, but have these huge cytoplasmic processes that reach up throughout the layers of the spinosum. This is a micrograph, a histology slide showing you melanocytes, so it's specially stained for melanocytes. Cell bodies of the melanocytes are these darker cell bodies, and then you can see these huge cytoplasmic processes that reach up all throughout the many, many layers of the stratum spinosum. So thick skin versus thin skin. So let's look at this cartoon picture here. You can read through this slide on your own. I'm just going to use the pictures to talk about what that slide says. So thick skin would be you would see pretty much all the same layers except for, so in thick skin, you definitely see a prominent stratum lucidum. And then look how thick the stratum corneum is. It's really, really thick. You would find thick skin, so this would be the stratum basale, stratum spinosum, stratum granulosum, that darker staining, then stratum lucidum, stratum corneum. You find thick skin anywhere where the areas of the epidermis that are going to be subjected to even higher amounts of abrasive forces and mechanical trauma. So the palms of your hands, the soles of your feet, you'll find that will be thick skin, so it will have a much thicker stratum corneum layer. If you notice over here in this cartoon image of the cutaneous membrane, thick skin will lack hair. So we don't, over here in thin skin, we see hair follicles and hair shafts and sebaceous glands. But in thick skin, we don't see hair. So if you look at the palms of your hands or the soles of your feet, you don't really have hairs there. But you will see prominent sweat glands. You know, your palms do get sweaty, especially if you're under some sort of stress or fear. Your palms can get a little sweaty. It's because you find sweat glands, but no hair follicles. Thin skin is going to be pretty much everywhere else. That's not the palms of your hands or the soles of your feet. The soles of your feet, you'll hear that referred to as the plantar surface. All right, plantar, that's the soles of your feet. So thin skin, we still see stratum basale, we see a spinosum, we see a granulosum, and this has a little bit of a stratum corneum, but it's much, much thinner, and it's kind of broken apart here, but much thinner than the stratum corneum over here. So thin skin is pretty much located everywhere else, and you see prominent hair follicles. So anywhere where you have hair, even fine little hairs like on your face, that would be thin skin, hair follicles, sebaceous glands, sweat glands, the whole gamut. All right? And so that is the